Good morning, one and all. Here we are in the fifth lesson called Transpiration. Let us have some introduction. Water is the most essential and abundant constituent of plant tissues. All plants absorb a large amount of water from the soil through the roots and transport it to the whole plant body. Though plants cannot survive in the absence of water, yet only a very small quantity of water, that is 2 to 5%, is actually used for the maintenance of turgidity, translocation of dissolved minerals and photosynthesis, etc. The great amount of water absorbed by the plant, if accumulates in the plant cells, can endanger their survival. This excess water, that is 90 to 98%, is removed from the leaves in the form of water vapor. So we can say transpiration is the phenomenon of loss of water in the form of water vapor through the aerial parts of the plant. And all plants absorb a large amount of water from the soil through their roots and they transport it to the whole plant body. So what happened? Though plants cannot survive in the absence of water, only a very small quantity of water, that can be 2 to 5%, is actually used for the maintenance of the turgidity, translocation and photosynthesis of the plant. Now the great amount of water absorbed by the plant, if accumulates in the plant cell, then it can endanger their survival. So what happens? This excess amount of water will have to be taken out. So the excess water, that means 95 to 98% of water, is translocated to the leaves and it is lost to the environment in the form of water vapor. So this process or this phenomenon is called as transpiration. Now transpiration is a very useful process for plants. First thing, it creates suction force in the stem to enable the roots to absorb water and the minerals or nutrients. Second thing, it helps for cooling of the plant in hot weather. And this process is demonstrated and studied by a number of experiments which we will study further. So, as we know that all plants continuously absorb water through their roots, this water is conducted upwards through the stem and is distributed to all the aerial parts including the leaves. And only a very small quantity, which I told you, around 2 to 5%, is used by the plant in the photosynthesis. And the rest of it is almost lost in the atmosphere as water vapor, and that is the result of transpiration. So here we can say that transpiration is the phenomenon of loss of water in the form of water vapor through the aerial parts of the plant. Let us see the steps involved in transpiration. Plants absorb a large amount of water from the soil through the roots. Second thing, water moves through the cortex, endodermis and pericycle of the roots into the xylem of the stem. Third step is, xylem forms a complex network of water which is a conducting tissue, water conducting tissue, and finally ends in the fine veins of the leaves. Contain a large number of living thin-walled mesophyll cells, and these are loosely arranged and have a large intercellular spaces. Water then moves from the veins into the mesophyll cells and evaporates into the intercellular spaces. And finally, the intercellular spaces gradually become saturated with water vapor and leading to their diffusion into the less saturated or atmospheric air through the stomata. Demonstration of transpiration. So first experiment we'll see, which will demonstrate the occurrence of transpiration in plants. So what are the materials required for this experiment is, take a healthy plant with a fresh broad leaves, a polythene bag and threads. So what is the method? Select a healthy plant with fresh and broad leaves, cover one of its branches with a polythene bag and tie its mouth at the base of a stem with the help of a thread. Leave the setup undisturbed under sunlight for 2 to 3 hours. Now take another empty poly bag, name it as B, and tie its mouth with a thread. Keep this empty bag also undisturbed under sunlight for 2 to 3 hours. Now what will be the observations? Water droplets will appear on the inner surface of the poly bag A, whereas the empty poly bag B does not contain any water droplets. So what is the conclusion there? In polybag A, the plant releases water vapor during transpiration, and these vapors condense on the inner surface of the bag and appear as water droplets. The polybag B does not contain any water droplets because of the absence of plant and the occurrence of transpiration, as we have seen in the previous class. This experiments which is again taken to see as a control. So this was the first experiment which will demonstrate you. The second experiment is to demonstrate that plants release water vapor during transpiration. See, what are the materials required here? Again, take a healthy potted plant, a soil filled pot, bell jars, polythene sheets, cobalt chloride papers and some wax. So what's the methodology of this experiment is, select a healthy potted plant and cover its soil surface with a polythene sheet to prevent the evaporation of water from the soil. Now, take a bell jar, label it as A and attach a strip of dry cobalt chloride paper on the inner side of the jar. Third thing, now place this jar over the potted plant and seal the rim of the jar with the wax to avoid the entry or exit of any moisture from it. Now leave this setup undisturbed for 2 to 3 hours under sunlight again. And what control setup we should take? Another potted pot filled only with soil but without a plant, and cover its surface too completely with a polythene sheet. Place this pot under another bell jar level B containing the dry cobalt chloride strip as in A, and leave this experiment also undisturbed for 2 to 3 hours under sunlight. What will be the observations? In the experiment, we'll see water droplets appear on the inner surface of the jar and the cobalt chloride paper turns into pink color. Whereas the control setup does not have any water droplets in the bell jar, and the color of the cobalt chloride paper also remains the blue. So what is the conclusion here? In the bell jar A, the plant releases water vapor during transpiration, and these vapors condense into water droplets on the inner surface of the bell jar, changes the blue color of the cobalt chloride paper to pink. Whereas absence of the plant and thus absence of transpiration in the control setup results in the lack of any moisture in the jar, and the color of the cobalt chloride paper remains. The color of the cobalt chloride paper is blue when dry, but it becomes pink when it gets moistened. So this was the experiment which was conducted to demonstrate the plant released water vapors during the transpiration. In your book, the same experiment is given but in a little different way. So let us see that experiment again. See, in the figure 5.2 on page number 56, there are certain arrangements shown, isn't it? So arrange 3 setups A, B and C. In setup A, take a small well-watered potted plant, preferably with 1 broadleaves. Enclose the pot completely within a polythene bag and tie the mouth of the bag firmly around the base of the stem. And this would prevent the escape of water vapor from the pot. Now cover the entire plant under a bell jar as seen in A. Now for setup B, arrange another similar plant and cover it with a bell jar exactly in the same manner as the first one, except that here you also keep a piece of dry cobalt chloride paper by the side of the plant inside the bell jar B. The paper may be pinned to a wooden stick or to a strip of a cork sheet. Third setup is without the plant but still containing a smaller piece of cobalt chloride paper, and all these 3 bell jars together are kept in the sun. And about half an hour, if we observe that the first bell jar A would show water vapor condensing on its inner walls, second bell jar would show a similar condensation, and at the same time the initially blue cobalt chloride paper in it would turn into pink. The blue color of the cobalt chloride paper in the third bell jar does not change at all, and there are no water vapors on the jar's inner walls either. So the third bell jar in this experiment is simply a control which proves that there was no moisture in the air due to transpiration, as there was no plant in it. And by taking the 3 bell jars as described above, there is a double visual proof of transpiration: condensation of water vapor into droplets and change of color in cobalt chloride paper. Our next topic is measurement of transpiration. There are a number of methods for measuring transpiration. Some of these are, our first method is weighing method. Here, a small lightweight potted plant can be weighed before and after the end of a certain period of time. The soil surface and the pot should be fully covered to prevent evaporation from the surfaces other than the plant. The loss in weight by the plant during that time is due to the loss of water by transpiration. An improvement in the weighing method can be made by using a glass bottle linked by a rubber tube to a graduated tube filled with water as shown in figure 5.3a on page number 57. The water level in the side tube falls to demonstrate loss of water through transpiration from the leaves. This would indicate that the volume of water loss that can be compared with the loss in weight with the help of a weighing machine or by converting cc into grams. That is, 1 cc of water weighs 1 gram. Now, another weighing experiment which is very easy to be understand is that it can again be done by using a test tube filled with water and inserting a leafy shoot in it and pouring some oil on the surface to prevent the loss of water by evaporation. You can see that on figure again 5.4. See, a test tube is there, water is filled in that, a twig is inserted and some drops of oil is also put on the surface of the water, and that oil is put on the surface so that it will prevent the loss of water by evaporation from the surface. Now place this test tube in a small beaker and weigh them together. Remove the intact test tube and keep it straight in the test tube stand for a few hours. Now weigh it again keeping in the same beaker. Any difference in the weight will indicate loss of water by the shoot, and that loss of water will be nothing but due to transpiration. Since there are no roots to actively absorb water, the water loss through transpiration will be much. Second measurement of transpiration, second method is our photometer method. Photometer is the device that measures the rate of water intake by a plant, and this water intake is almost equal to the water lost through transpiration. See, there is a note in a blue box. Photometers are of various types designed by the various scientists to study and measure the rate of transpiration, namely Farmer's photometer and Ganong's photometer. And these photometers are named after their scientists. So, which helps to measure the rate of water intake by a plant. Next is our Darwin's photometer. This also helps to demonstrate the suction force created due to transpiration, and Gary's photometer demonstrates unequal transpiration from the two surfaces of a dorsiventral leaf. So, different photometers are there for different measurements. So, here we will go through Ganong's photometer, which is only there in our syllabus. So, in a Ganong's photometer, here you can see the device. A twig of some suitable plant, example you can take of a Collier's plant, cut with a sharp knife and fixed it in a apparatus called as a photometer, which is shown in the figure 5.5 on page 57. The entire apparatus is filled with water so that no air spaces are present. An air bubble is introduced into the horizontal graduated capillary tube, which is dipping into the beaker containing water. This is done by lifting the bent capillary tube above the colored water so that air may be sucked in due to suction pull and is again dipped into water. And this is done only to introduce a air bubble. Now, as transpiration proceeds, that is the loss of water from the twig, a suction force is set up which pulls the water from the beaker and the bubble in the capillary tube moves along. The readings on the capillary tube would give the volume of water lost in a given time. The air bubble can be brought back to its original position by releasing some water vapor from the reservoir into the capillary tube by opening the stop cock. And all this you can see if you see the experiment or figure. You can clearly understand how this air was inserted inside the capillary tube. See, here it's written, photometers do not measure the water loss during transpiration, but it measures the water uptake by the cutting shoot. And some of the water is used by the cells to carry out other process also. For example, in the process of photosynthesis. Precautions in the use of photometer. The photometer should be made completely watertight. The twig should be cut obliquely to allow the large surface for the water intake and underwater to avoid suction of an air bubble into the twig, which will stop the absorption of water into the xylem. These are the two precautions which we should take while using the instrument called photometer. Use in the photometer. So, introducing the air bubble is not so easy. The twig may not remain fully alive for a longer time, and any change in the outside air temperature may affect the position of the air bubble in the capillary tube. So, these are some of the limitations which you should take care while performing the experiment on the photometer. Kinds of transpiration. Transpiration from the aerial parts of a plant occurs from three different reasons. First of all, through the stomata which are present on the leaves. So, it is called as stomatal transpiration. Second is directly from the surface of the leaf and stems, that is called as cuticular transpiration. And third is from the lenticels, which are the minute opening on the surface of older stems, and they are called as lenticular transpiration. The major part of the transpiration occurs through the stomata, whereas the other two types of transpiration contribute to a very mechanism of stomatal transpiration. Stomata are minute opening on the epidermal layer of leaves, and stomata is surrounded by two bean-shaped guard cells. During daytime, the stomata are wide open primarily for the intake of carbon dioxide for the process of photosynthesis, and the number of stomata may range from 1000 to 10,000 per cm square. In stomatal, the water vapor escapes through the stomata of the leaf, and water, after absorption by the roots from the soil, rises up through the stem and reaches the tissue of the leaves through the veins. A large number of spongy mesophyll cells in the leaves have their surfaces exposed to the intercellular spaces, and these surfaces of the cells gives out some of the water as a thin film. The water from this thin film evaporates and the water vapor formed saturated in the air in the intercellular spaces. The water vapor then diffuses into the other connecting intercellular spaces and finally reaches the substomatal space, from where it escapes through the stomata. The entire movement of water vapor from the surface of the cell into the outside atmosphere is a result of diffusion, which is shown in figure 5.7. The molecules of water vapor, like those of any gas, move from the region of the higher concentration to the region. Figure 5.8a shows the movement of water through a leaf. The cell sap in each cell exerts a turgor pressure outward on the cell wall, and this pressure forces some water out of the cell wall into the airspace between the cells. Here the water vapors evaporate and the water vapor diffuses through the airspace between the mesophyll cells into the substomatal space, from where it finally goes out by diffusion through the cells that lose water in this way. Replace it by drawing more of it from the nearest vein, and most of this water travels along the cell walls by the process of imbibition, and only a small quantity enters the cell by osmosis, which is shown in figure 5.8b on your page number 59. Thousands of leaf cells evaporate water in this way and causes more of water to be pulled from below the xylem vessel. The transpiration pull thus creates or it can draw up water to about 50 meters or more in tall trees. This is the reason how the tall trees are able to absorb water from the roots till the shoot. Now, more transpiration occurs from the undersurface of a dicot leaf. See, there are small or more stomatal openings on the undersurface of a dicot leaf, and therefore more transpiration occurs from the undersurface. This can be proved by an experiment in which pieces of dry cobalt chloride paper are attached to the two surfaces of a leaf and held in the position by two glass slides on either side, tied together by a elastic band or held by the clips. The leaf should remain attached to its own plant. The piece of paper which is facing the upper surface of the leaf either does not turn pink or will turn into pink in a much longer time than the one on the lower surface, which turns pink much faster comparing to the previous one. This proves that more transpiration takes place from the lower surface, which is an account of the numerous stomata found on it. So this proves that the dicot leaf is having more stomata in their undersurface. So this is how we can show the experiment to demonstrate the different magnitudes of transpiration from the two surfaces of a dicot. So for today's topic, this much only, and I would see that you should do all the progress check from the textbooks and go thoroughly the lessons. And the next topic, that will be the factors affecting the transpiration in the next class.